Happy Valentine's Day! To celebrate, here are some fun facts about the amino acid valine. Feel free to bring them up on your date tonight if you need conversation starters.
So valine is one of our amino acids or protein building blocks. There are 20 common ones of these, and different numbers and combinations of them linked together to give you these long chains of amino acids called polypeptides that then fold up to give you functional proteins. So these amino acids all have a generic backbone part that allows them to link together, and then a unique side chain or R group that sticks off, kind of like charms on a charm bracelet. And so the different amino acids have different like charms, and these have different properties that are going to affect how the protein folds and functions. So the protein is going to like fold up so that it accommodates all of those side chains the best. And then it takes this form that's going to affect its function, kind of like a spoon versus a fork versus a knife type of thing. Valine has these properties that's going to affect the proteins that it's part of. And so one of the main properties about valine is that it's hydrophobic. And so what this means is that water doesn't like to hang out with it. And so I have a longer post on valine, by the way. This is just like the brief version in honor of the holiday, but much more in the other post. This is just like a quick facts edition. So if you want to know more, I go more into depth about why on these things on other posts. But basically hydrophobic means that water doesn't want to hang out with it. It has to do with like this unequal sharing of electrons, making parts of water partly positive and parts of them partly negative. Similarly, some amino acids have parts that are partly negative and parts that are partly positive. But with valine, you have carbon and hydrogen, which share fairly, so we call it like nonpolar. So basically, this doesn't offer anything that the water wants, and so the water is going to exclude it. And this is going to force the regions that contain valine tend to be towards the center of the protein. So all the water is kind of like cinching up around it and forcing those parts into the center of the protein. This can be a problem then if you have a mutation that puts this hydrophobic residue onto the surface of the protein. And in fact, this is what happens in sickle cell anemia. So sickle cell anemia affects this protein hemoglobin, which carries oxygen through your blood. And there's this surface residue. So a residue is what we call like an amino acid once they've been linked together. And so there's this glutamate here, and glutamate is negatively charged. Valine, and so negative charge, like water likes that, that's hydrophilic. Valine, hydrophobic, no charge. And it's out here on the surface, and now the protein's not going to be happy. And so what's going to happen is that, especially under depending on the oxygen concentration, and this hemoglobin is going like through all of these low, high oxygen, and it's like giving and taking oxygen, and all of these things, and it's stressing out this molecule. And now you've got this hydrophobic acid amino acid on the surface. And what's going to happen is it can find like another like hydrophobic pocket on another hemoglobin molecule, and it links up into the form of these long fibers and twists together into these weird structures. And this is happening inside of the blood cells, and it's going to cause the blood cells to adopt like a sickle shape, and then clump together in like clogged blood vessels. And so this is why you get problems with sickle cell anemia, and it has to do with the hydrophobicity of this valine. So you're not going to commonly find valine on the surface of proteins, and you're also not going to commonly find it in alpha helices. And so alpha helices are one of the main types of secondary structural motifs. And so we can talk about different levels of protein structure. So you have the primary structure, which is the order of amino acids, the secondary structure, which involves interactions between the backbone, so that generic part of the amino acids, and then the tertiary structure, where you have the side chains playing a role, and then quaternary, if you have like different chains, like so in hemoglobin we have like these different chains, so it has like a quaternary structure. But here we're talking about the secondary structure, and this involves like these beta plated sheets and these alpha helices. So these are coming from interactions, these like hydrogen bonding between the backbones, between like these nitrogens and these hydrogens. But because different proteins have the same like linkages, even if they have different side chains, but that backbone part is the same. So you might expect that they all form these same secondary structures. And so this is why you see these beta strands, you see these alpha helices a lot, but it turns out that the structures you can form, they're restricted. You don't see them with all amino acids. You see them with like certain combinations. Certain amino acids are more likely to be found in those, certain aren't. And this has to do with kind of like the bulkiness and stuff of those amino acids. So it's harder to turn around if you have a lot of bulk near your surface, or if you have something big and bulky. So valine doesn't look that big, but because it has this branching, it has this branch point here. So it has this V. This is how I remember the structure of valine, because it looks like a V. That's not how it got its name. But anyway, it's branched close to the like to the chain, the backbone. And so this is going to make it hard to rotate around. So you can't rotate around like the actual peptide bond, but you can rotate on like either side of it, because this is partial double bonded character, which is part of the reason why these bonds are so strong. But around here, you can rotate a little, but how far you can rotate is going to depend on the side chain. And so there are different angles that are preferred in like a helix or a strand. And so valine, it is hard for it to form these helices because it has this bulk near its surface. And so actually leucine, even though it's bigger, it has its bulk farther out. So it's easier for it to be, take on some weirder angles. Speaking of leucine and valine and their branchedness, also isoleucine is one of these branch ones. These are what we call branch chain amino acids. And the consequence of this is that they go through some kind of similar breakdown pathways, and they can, so like metabolic disorders that interfere with these pathways can affect the breakdown of multiple ones of these, depending on where you get a mutation. So something else about valine, and so when you break down valine, what you end up with is you get the succinyl-CoA, which can go into the tricarboxylic acid cycle, sometimes called the Krebs cycle or the citric acid cycle. And this can be used to make glucose. And so we call valine glucogenic. And we can do things with this breakdown products, but we can't actually make it. So it's an essential amino acid in the dietary sense, meaning that we have to get it through our food. We can't make it ourselves. And this is true for all of the branch chain amino acids. As for the discovery, final note, so this is actually it was actually kind of like discovered in 1856 by this chemist Bessenes, and he found it in pancreas extract. He was able to figure out like the empirical formula, so like what number of each type of atom was in there, but he didn't know how those atoms were connected. And then other scientists found out more information. But they still couldn't figure out like the actual structure, like how were those things like actually linked up. And then finally in 1906, there were separation techniques that could separate the different like stereoisomers, so different arrangements in space. And then they figured out the structure, and then from thenceforth, valine would refer to alpha amino isovaleric acid. And so yeah, that's where the name so the valine comes because it's like valeric, it looks like valeric acid. So one last thing, fun fact you can bring up tonight. So the L and the D stereoisomers of valine, so kind of like the left and right-handed versions. So our bodies only use the L version, but apparently the L and the D taste different. So don't taste things in your lab, but this guy Fisher did. And he found that the D and the L versions tasted different. D-valine is bittersweet and L-valine is all the way sweet. And so our bodies use the sweeter form of valine, which is also in keeping with today's topic and today's festivity. So hope you have a great day. Whether or not you have a romantic loved one to share things with, just have fun with your friends, and you always have molecules you can hang out with.